I did notice that there are a few students in the comment section asking about phloem structure and function. And to be fair, a lot of students who I do teach in face-to-face classes also request that I go into detail about the phloem structure and function because it's quite confusing actually, isn't it? So we will start by describing what exactly is the phloem. So the phloem tissue is categorized as vascular tissues. And why do we call them vascular tissues? Because they contain tube-like structures. But the tube-like structures in the phloem actually transport sucrose and amino acids. These are the two main things that phloem transports inside the plant. Where are the location of the phloem tissues? As you can see here, I'm showing you the root cross section and the stem cross section and the leaf cross section. TS just means transverse section, which means cross section, same thing. Where I have highlighted in orange or brown or beige, I have no idea what that color is. Looks beige-ish, okay? That's where the phloem tissues are actually located. So you have to be mindful as to the location of the phloem in the roots, the stem, and also the leaf. At least that's the first thing we have to know. So the thing we have to talk about then is, what is the phloem? The phloem, while it is described as a tissue, it is not just a single thing, but it's actually made out of two different types of cells. And the two different types of cells are the companion cells and the phloem sieve tube element. Some books will just refer to it as the sieve tube element, but both are accepted in the exam. So let's talk a little bit about the formation of the phloem. Again, good news, you don't have to memorize this, but it's good to understand where the phloem comes from. So remember, the phloem is made out of two different types of cells, and the black color cell is a companion cell, and the orange color cell is the immature or the young phloem sieve tube element. And notice that the companion cell and the phloem sieve tube element, they are connected by something known as the plasmodesma in the middle. That's the cytoplasmic connection that they have between the two. So what I'm doing here is I'm going to show you a few companion cells and a few phloem sieve tube element. And what actually happens is, notice what happens to the orange color cells. Those orange color cells, the cell walls will start to break down, all right? That's normal stuff, but it doesn't break down completely like the xylem vessels. There are still like some cell walls that remain in place. So, and then some of the cytoplasm and the organelle will also start to drain away within the sieve tube element. And that's how the phloem sieve tube is actually formed. The phloem sieve tube is made out of many sieve tube element cells joined together. And that's the tube. The orange color section over there that I'm circling is actually the sieve tube where transportation takes place. But the phloem itself is actually made out of two things. The phloem is made out of companion cells connected to the phloem sieve tube element through the plasmodesma. That is something very important that we have to know. So when I'm just zooming into one of the companion cells and the phloem sieve tube element, we notice that the companion cell still has important organelles, like it has the nuclei, it has vacuole. It may also have organelles like mitochondria, rough ER, Golgi apparatus and such. But look at the phloem sieve tube element. It does have a little bit of cytoplasm there, as I'm just showing you through the arrow, but the volume of cytoplasm is almost very negligible. It is important to have some cytoplasm, obviously, for the phloem sieve tube element. I'll explain why, but the point of the matter is the phloem sieve tube element will not have a lot of cytoplasm. And some students will ask, does it have any organelles like mitochondria? It may have like a few mitochondria, but it usually does not have nuclei and other large organelles. Another very important thing to know about the phloem sieve tube element is the parts where I'm highlighting, where the cell walls are broken down into those tiny pieces, and those are referred to as sieve plates. And again, I'm just circling over that. The companion cell and the phloem sieve tube element are connected via the plasmodesma. So if you still can't imagine this, let's try to draw this out in a slightly different diagram. Remember, we are looking at this through a longitudinal section. Now, if I were to highlight that yellow strip over there and look at it through the transverse section, transverse meaning to say we look at it from the top, what we will see is the companion cell with the vacuole, and we will also see the sieve tube element where they are connected through the plasmodesma, and the sieve tube actually has some cytoplasm in there. But if I were to highlight that particular section, and if I were to view the phloem through the cross section in this area, how it will look like is it will actually the companion cells still look normal because I can see the nucleus, but where I'm highlighting, they are not connected through the plasmodesma. So how it will look like is you will actually be able to see two different cells. How does the sieve plate actually look like? The sieve plate looks like a cheese grater, doesn't it? And the sieve plate just has those tiny little holes to allow transportation to happen. That's what the sieve plate actually looks like, by the way. So the sieve plate is just the broken pieces of cell wall. It's not totally broken. There are some tiny little holes that forms the sieve plate. If you still can't imagine this, let me try another method. I'm going to do a three-dimensional view of the drawing. So I'm drawing out the companion cell in black, and the orange color cells, as you can see there, quite elongated. I'm going to make it a bit more three-dimensional. Those are the phloem sieve tube element. All right, so I'm drawing out two phloem sieve tube element, and those and you can see the companion cells. It has cytoplasm. They are connected to the sieve tube element via the plasmodesmata, and the phloem sieve tube element has a little bit of cytoplasm, not a lot, but it has a little bit. Okay, and what is very important to know, those highlighted parts are the sieve plates. So this is just a three-dimensional diagram of how the phloem sieve tube looks like. It's not the best diagram I've drawn, but it kind of gives us an idea of what the phloem is all about. So long story short, the phloem tissue is made out of two types of cells, companion cell and the phloem sieve tube element, and the companion cells and the phloem sieve tube elements are joined together through the plasmodesma. That's the first thing we have to describe when we are talking about the structure of the phloem.